Great. Thanks to Randy and Brian, Dimitri, Sarah, David, especially Sarah for all of her help in herding us cats into doing what we're supposed to be doing. I've really enjoyed the meeting. So after Laurel's talk, I was thinking I really should be talking about normalization models and cascades of normalization models, and I was tempted to switch my topic instantaneously, but I've decided not to. So you're going to get what I advertised. I'm going to talk about some work that I've done. It actually started quite a few years ago, and it's ongoing, although I'm waiting to find another student to dive in on this. The student who did most of the work I'll tell you about is Deep Ganguly. The name may sound familiar to you because he is the brother of Surya Ganguly, who's a computational neuroscientist at Stanford. Deep was a graduate student in my lab who went on to a startup and is now working for the Chan Zuckerberg Initiative in Palo Alto. So much of the work in my lab can kind of be described with this simple diagram. I'm really interested in understanding how sensory representations are formed based on properties of the environment, how those things are represented in the brain and the physiology of the brain, and then how those things lead to what we would call perception. And I want to understand all of that, which is a little bit ambitious. To keep it a little bit focused, I concentrate on vision and also a bit of work in the auditory system. And we can think of this process as it's artificially divided here into two stages, two steps, if you like. Think of it as an encoding step and a decoding step. By the way, is there a way to turn off these blackboard lights?
Yeah.
They're very annoying.
Really cool.
I think it's at the console.
At the console. Oh, look at that. Yeah, that's better. Thank you. Okay. Is that better for you as well?
Yeah, it's worse for Chris, but it's much better for us.
Okay, terrific. Well, I'm happier. Okay. Okay, so we often artificially divide this process into two steps. You can think of it as encoding and decoding. Encoding the mapping from the input, the environmental input. In my case, I'm going to be talking about mostly about vision, into internal physiological representations, populations of cells, spikes, and then decoding, which is the transformation from those populations of spikes into what we would call perception for the cases I'll be thinking about, but it could be all the way to motor activity or to memory. If Randy were up here talking, he would be talking about memory. But we can think about, I'm going to think about encoding and decoding, and I'm going to think about normative principles that we have to sort of structure and conceptualize these two stages. And in particular, the main normative set of normative principles that we use for thinking about encoding are efficiency. This really comes from Barlow and Atneve and others who talk about the idea that sensory systems have to use their resources in a reasonable way. Those resources are limited, and so they have to find ways to take all of that information that's flowing, for example, with your eyes, and compress that and squeeze that into some representation that makes sense and that doesn't cost too many spikes and doesn't take too many neurons. On the decoding side, there's optimal inference, basically Bayesian inference, that is, given a pile of spiky neurons, how do you figure out what it is that's out in the world? You have a noisy partial representation. I'm going to slightly disagree with Laurel about being able to reconstruct the world from complex cells. I don't actually think that really is true, at least not in the brain. But in any case, the idea is that you have some partial noisy representation. What is it you can extract, and how do you go about doing that? What does it mean to have a percept or an understanding of what's out in the world, given that partial representation? Okay, so I'm going to actually talk about both of these things, but the first 3/4 of the talk is just about the first one. Okay? And for today's talk, I'm going to really strictly limit this down to a very simple case. I'm going to think about one variable at a time, so I'm going to imagine there's some variable out in the world called S. That's some sensory variable, and we're going to think about the probability distribution of that as it occurs in the world. For the physiology, I'm going to be thinking about either one cell or a population of cell, the kind of things classically that were recorded with extracellular electrodes, single responses to these stimuli, and we'll think of those as tuning curves. Okay, so this is a response of one cell, for example, in B1, to stimuli drawn from along this axis. We can think about orientation tuning as a prototype example, but it could be anything. This S can be any variable that we want that has to do with real-world signals. And I'll think about a population of those. And then on the perceptual side, I'm just going to think about something, again, that's very simple and very traditional, something that we typically measure. When we measure perceptual capabilities in terms of the variable, let's say S, we often do things like measure the discriminability, we measure detection capabilities, we measure the ability of humans or animals to tell the difference between two stimuli that are near each other along this axis. And we measure things that often take the form of something like this. This is a psychometric function, for those of you that know about this. This is the fraction of time that a subject would say that, let's say, the red stimulus presented here is reliably detected. The fraction of the time the person would choose the red stimulus if you ask them to choose which one is bigger along this axis. And so as the stimuli differ more and more, the answers go toward 100% or 0%, and in between there's a 50% point, which is the sort of point of subjective equality where you can't tell the difference between the stimuli. Okay? So we would measure often the discriminability of these stimuli by measuring the slope of a function like that fit to data from human observers. That would tell us something about how easy or how precise the internal representation is. And I'm going to come back to this as a measure of how populations can lead to this behavior and how efficient populations can explain the kinds of behavioral observations we have in these kinds of psychophysical data. Okay, so I'm going to go through, this is the main question I'm going to ask. It's a pretty simple question, and it would seem like it should have an obvious answer, but at least when we started, it wasn't so obvious. And the answer came out a little bit surprising, at least from my perspective. Okay, so the question goes like this. If you have some environmental stimulus variable S, it's again, just if you want a concrete example to keep in mind as I describe this, think of orientation. Okay, so local orientation measured in some scene, it has some distribution. It turns out it's not flat. It actually looks kind of like this. There's very high preponderance of verticals and horizontals in our world, basically because of gravity and the fact that when we're looking at the world, we tend to keep our head more or less straight up and down. So there's lots of verticals and horizontals, fewer diagonals, if you like, or off vertical or horizontal. And the question is, if I give you n neurons that are tuned for this variable orientation, and I tell you that the number of spikes you're allowed to fire in this population is limited, let's say to some average firing rate R, say two resources, the number of cells and the number of spikes, and I'm going to give you an allocation of those resources. And now the question is, how do I distribute the cells? Where do I put them along this axis? How do I lay them out so that I can transmit a lot of information about what's out in the world, this variable S, and what are the perceptual implications of that solution once we've figured out what the answer to the first question is? What does that mean in terms of perception? Okay, so, and you know, these are just examples, so it might be, maybe the right thing is to lay them out uniform like this, a uniform population of cells that have the same tuning width and the same basic shape, and they're distributed along the axis. Or maybe I should make narrower tuning curves in the center where the probability is higher, where I'm more likely to see stimuli, and make them broader in the diagonals. Or maybe I should make the firing rates higher in the center and the firing rates lower in the diagonal. Or maybe the other way around. Maybe since these things in the center occur frequently, I want the firing rates to be lower because those are costing me a lot of spikes every time they occur, and if they occur frequently, then I'm going to be burning a lot of my metabolic budget just trying to code things that are sitting at the verticals and horizontals. So the question is, you know, what's the right answer? Or can we get a right answer? Is there enough of a constraint on this problem for us to actually achieve an answer, a unique answer that tells us how to allocate the resources? Okay. So as a warm-up, I'm going to try to stay away from math. I'll show you a tiny bit of the math just so you have a flavor of what's involved, but I'm going to leave most of it out. I'm going to try to show mostly pictures so you get a flavor of what the solution looks like and what we go through to solve this. And to sort of warm up, let's think about a homogeneous population. This is something that's been studied by many people. There's a lot of literature on it. Imagine we have an oriented stimulus. We have a homogeneous population like this. The way the stimulus gets encoded internally is in the responses of some of these cells, and in this particular toy picture, there would be 3 response levels, 1, 2, 3. These 3 cells that are responding. The rest of these cells would not respond to this stimulus. They're just be sitting there, maybe running at background rate or not firing any spikes at all. And from that, we have to decode that and produce an internal perceived concept of orientation. And that's the thing that our judgments would be based on if we were asked to make decisions or to make motor actions based on that percept. Okay, so of course, this is noisy. The process is noisy. The cells don't respond the same every time. And if we ran this multiple times and we measured from these cells, we would see that each of these cells would have some fuzzy cloud of possibilities, a probabilistic cloud, if you like, around them. And the actual responses would not lie on those tuning curves. They would be somewhere near them. And on particular trials, we would get different answers. And at the human subject level, we would also get different answers. If we were asking people to discriminate stimuli, we might get different answers on different trials, even showing exactly the same stimuli. Okay, so the variability, the accuracy here is limited by that noise, that variability in response. We don't have to call it noise. Maybe it's not stochastic. Maybe it's coming from things that we have not controlled or things that we have not measured, but it doesn't matter. It just doesn't matter. Whatever it is, it's something that doesn't tell us about the stimulus, and therefore it's kind of getting in the way, and it's causing fluctuations in variability across trials. Okay, so lots of people have studied this problem, and we know that the accuracy is limited by the noise. And we know that, for example, because of the properties of neurons, that they're sort of Poisson-like, they become more accurate or more precise if we boost their firing rates. So if we take the same population that was giving us this accuracy and we double all the firing rates, well, it'll get more accurate. That is, our percept should get more accurate. And we can roughly calculate how that should work for a Poisson process. So that tells us that adding more resources, i.e. more spikes, gives us a more accurate representation. Similarly, if we add the other kind of resource, more cells, so if we double the number of cells, it's the same shapes, tuning curves, just twice as many, now we get more responses. And it's just kind of, the idea is very simple. Now we're just going to average over more cells. We have more noisy responses, and that also will give us a more precise representation, a more precise estimate, if you like, of the underlying orientation. Okay, so we can increase the number of spikes, or we can increase the number of cells. Either one of those is going to give us an improved representation. And it turns out we can, when we increase the number of cells, we can do something else. We can also make them narrower. Because maybe if we keep the same width, they're overlapping more if we double the number of cells. And so maybe we can do better if we actually also reduce their widths, keep them shoulder to shoulder, the same relative spacing as they get narrower. And it turns out that does help, and we'll get an even bigger boost if we can do that. We get a bigger boost from increasing the number of cells if we kind of match their widths to their spacing. Okay, so that's just to give you some intuition. And now we can go back to the real problem. The real problem was not to deal with homogeneous populations. On the contrary, what we want to do is figure out how to lay them out in general. They can be irregular, as is illustrated here. And we want to know what's the best solution given the distribution in the world. So we can work through this. Now we're going to show you a little bit of the math. If you don't want to see the math, close your eyes for 2 slides and then come back. So here's how it goes. We're going to assume independent Poisson processes. It's a very standard and very simple model. It's the easiest thing to work with. It is I can give you all kinds of reasons why we should use this, and I can also give you lots of reasons why we should not use this. I think that it's good enough for our purposes in this project. So we're going to stick with it. This will generalize, it turns out, to exponential families, and you can even put correlated firing in, but I'm not going to talk about that here. Okay, so that was the first thing. We're going to assume independent Poisson. Second thing, we're going to assume, we started out trying to solve this problem with an arbitrary population of cells, figuring out arbitrary tuning curves and shapes, and it was a mess. It's a really difficult optimization problem, highly non-convex, lots of local minima. We couldn't figure out a way to solve it. Maybe somebody else out there who's a super expert on optimization can, but we could not figure it out. And we slowly came to a realization that the right thing was to constrain the problem with what we kind of knew made sense. And so what we're going to do is we're going to parameterize the population not by allowing arbitrary tuning curves. We're going to force them to be essentially a warped version of a homogeneous population. Okay? So the idea goes like this. We're going to assume, we're going to parameterize the whole thing with a continuous function, not discrete entities, a continuous function. We're going to call that little d. It stands for density. It's the density of tuning curves, that is how tightly packed they are. And the way we're going to implement this is, we're going to take a homogeneous population over here on the vertical axis. It's turned on its side, okay? And we're going to run them through this nonlinear warping function. This warping function is the integral of the little d. Here's the little d. Here's the integral. It's like a sigmoid, okay? And this warping function is just going to stretch or compress the axis in order to take this population and turn it into this one, okay? So here, where it's steep, these things get compressed. Here, where it's shallow in the two ends, they get stretched out. So we're going to take this population and stretch or compress it in arbitrary places, and we can, of course, according to this little d function, we can make it really dense and compressed. We're very stretched out in arbitrary places. We can do whatever we want. But this parameterization enforces the fact that the cells are going to always have the same amount of overlap. They're always shoulder to shoulder, okay? All the way through. These cells start out shoulder to shoulder. They're going to end up shoulder to shoulder after the warping because, of course, the same warping function is applied to every cell. Okay? So it's a homogeneous, it's a warping of the entire space, and everybody gets the same warping function. Right? So we're building that in as an assumption. It's not something that we're going to derive from the data or that's going to be learned from the model. We're assuming it right from the outset. But that's what's going to allow this whole thing to work and to be optimized. Okay, so that's one thing. And if you like, the right way to think about this is given the resource of the number of cells, you hand me the number of cells n, this is the thing that allocates that resource. Okay? This little function, little d, is going to take that resource and decide where to put it. Okay? When little d is high, we put a lot of the resource, a lot of cells. When little d is low, we have very few cells. They're very spread out. Okay? Okay, the other resource was firing rate, spikes. So we have another resource allocation function. We're going to call it G for gain. And that's going to tell us where to put the spikes. And that's also continuous. And we're going to just multiply the entire population by that function. And if that function is high, then the spike rates are high. If that function is low, the spike rates are low. And that's going to allow us to distribute the spikes. Where do we put them? Do we put them in the high probability regions of space or the low probability regions of space? Or just leave it uniform? Okay, so that's the parameterization. We have two functions. And the resulting tuning curves, the nth tuning curve will look like this. The nth tuning curve is a function of the stimulus S. It's going to be the G function, that's the gain, multiplied by some generic H function, which is just some smooth shape, that's been warped by this capital D. Okay? And then shifted appropriately for the nth cell. Okay, so the math actually is pretty simple. That's the whole population described right there. Everybody's warped by the same D. Everybody has the same G. And our goal then is going to be to optimize D and G. And here's the last thing in the formulation. We want to optimize them to transmit information about the environment. We're going to use, instead of Shannon information, we're going to use a bound on Shannon information. Don't worry about the details here. We're going to use the Fisher information, which is a second-order approximation to the sort of variability of the decoded, if you like, a decoded version of the orientation, or the variable S. And we're going to use that as a bound on the actual Shannon mutual information. Okay? And it turns out the Fisher information in our problem is very easy to calculate. It's proportional to d squared g. It's kind of interesting. So d counts twice as much as g. So increasing G is going to increase information, and increasing D, the density of cells, also is going to increase the information. And D counts twice, and the intuition is to take you all the way back to what I showed you about the homogeneous population. D counts twice because you use more cells, and you also make them narrower. And both of those things together is going to make it essentially twice as effective at transmitting information. Okay, so D squared G is the Fisher information, and here's the thing we're going to optimize. We're going to maximize the average of the log of the Fisher information. That's what we get for the bound on mutual information. And we want to do that so that the two constraints hold. So the constraints are that the integral of the density adds up to the number of cells we have. This just says we're supposed to have n cells, so this little function has to integrate properly. And this one says that the total spike rate, if we integrate G against the probability of occurrence of the stimuli, this is supposed to add up to the total firing rate R. Okay, so these are our two constraints. Remember, we have two resources, N and R. We're allocating them according to D and G. So these are just statements that the allocation has to be correct. That is, it has to add up to the right numbers. And then this is the thing we want to optimize. Okay, so amazingly, when you write all of that out, and you put in those approximations in that formulation, not only does it get easier to optimize, you don't even need a computer anymore. You just write down a page of math, and you go through some Lagrange multipliers, and you do some calculations, and out pops the answer, and there it is. The answer is that the density of cells should be proportional to the probability in the world, proportional with a fixed proportionality factor, which is just the number of cells. Not only that, the G is constant. So all the cells have the same maximum firing rate, and you put more cells into places that are high probability. So more cells with narrow tuning in higher probability regions of the space, and all the cells have the same gain. So the distribution, it's kind of surprising, and it seemed when we first did this, I said, okay, that's cute, we get an answer, we can actually do it without a computer, I like that. I actually did it with a pencil and paper, which was a pleasure. And I should have done it, really to emphasize that I should have waited until we had a power outage. And just sat there with a candle. That would have been really satisfying. Simple things in life. Okay, so the cool thing is, and now the real tests are, does it work? Does it actually predict anything in physiology? So we have a physiological prediction here, which is that the cells should have this kind of distribution. And does it work in terms of perception? So if we go and we ask for humans or animals or whatever, if we take perceptual measurements of discrimination thresholds, it turns out, oh, I didn't tell you this, right? So here's 2 more points. I told you all the neurons have the same gain. It turns out that if you calculate the mean response of every neuron should be the same in this population. Because the mean response is basically to integrate the probability of occurrence of the stimuli against the tuning curve, and that gives you the mean response, the expected response. And remember that the tuning curves are getting wider. They have the same height, but they're getting wider in regions of low probability, and they're getting narrower in regions of high probability. So and they do it proportional to the probability, or inverse proportional to the probability. So if you multiply the probability by the tuning curve and you integrate, you get a constant, because as the probability goes up, the tuning curve gets narrower. You carve out a smaller chunk. So the simple way of saying it is, every cell in this population is carving out an equal-sized chunk of probability from the world. Okay, so it's a little bit like those old pictures of the homunculus, these pieces of brain that are allocated to different parts of your sensory epithelium, you know, or your the touch system, right, or various other sensory systems, and you know the allocation is very uneven in terms of actual area of your body. So it's sort of why is that? Why is the allocation distributed the way it is? And in this theory, the idea is that every one of these neurons is going to carry an equal load. It's going to have the same mean response, and it's going to have it's going to carve out an equal chunk of probability. Okay. The other thing is that when you go through and you calculate the Fisher information, that actually gives you a very tight bound on perceptual discrimination. So now we can make a prediction about what happens perceptually, and that prediction, if you work through the math, looks like this. So you said that the perceptual thresholds for detection, for discriminating stimuli, should look like this. They should be some constant alpha, which depends on experimental details, divided by something that depends on the rate and the number of cells, and divided by the probability distribution. So the cool thing here is that all these constants are entering here, but in the end, this is just a function of 1 over the probability. In other words, high probability things should be more discriminable, low probability regions of the space should be less discriminable. So it's a simple prediction, and it's easy to go and test. Okay, so now let's look at some examples and see if it works, and you can decide for yourselves if it works. So I'm going to show you 5 examples. 3 of them are visual, 2 of them are auditory. I'm going to have to make some arbitrary choices when I do this. I'm going to have to choose what is my variable S, what population of cells do I think codes that variable S, and that's a little bit, iffy. And I'm going to have to then ask what do psychophysical behaviors look like on that variable S. The psychophysics I'll show you is all human. The neural responses are all going to be animals. They're going to be cats and monkeys. And I'm going to have to choose some area, some collection, some population of cells to look at. Still, well, you'll see. I think it works pretty well. So here's a variable, spatial frequency. We're going to take a large collection of photographs, 3 different databases actually. We're going to measure spatial frequency. It's pretty well known that spatial frequency in natural images falls kind of 1 over f. It's like a modified version of 1 over f. that's what this is. This is a power law, slightly modified. You can probably barely see it. There's a line under there, which is what comes from the data, a light gray line. There's a black line, which is a fitted model, which is a modified 1 over F model, and the fit is really good. That's why you can barely see the gray line underneath. It's a lot of data. Okay? It's a lot of images. Okay, so that's nothing about biology in that. That's just photographs. So here's the biology. So we take data from, this is anesthetized macaque monkey, D1. These are a very large set of recordings from James Kavanaugh and Tony Lofshin's lab. And what we're going to look at are 2 different plots. On the left is the frequency of, it's a histogram, of the number of cells that were found in this data set that had a particular preferred frequency. Okay, so this is the distribution of cells in terms of their preferred frequencies. The black line is the prediction that came from the photographs. The gray bars are the actual data. You can see that it sort of follows the black line. It's not following it exactly. And in particular, the deviations, I guess I would say that there's not enough at the very lowest frequencies, and we're kind of missing out some things at the highest frequencies. It's important to remember that when physiologists record from these neurons, they often go in and they find the neurons, they're driving a single electrode, and they're kind of playing around with stimuli to try to listen, literally listen, because they pipe it through an amplifier and listen to it on a loudspeaker. They're listening for cells, and they often are finding cells based on whatever stimuli they happen to be showing while they're doing that search. So what stimuli do they show when they're doing that search? They show stimuli that are kind of in the middle of the range that the cells tend to prefer. And so I think there's a little bit of a bias in terms of the collection of cells that one gets in a procedure like this. And that's my explanation for why our theory didn't perfectly predict data. I know none of you buy that, but I had to give you my story. Okay. This one is a little bit less subject to playing around and biases in data collection. This is the actual bandwidths, the width of tuning of the cells as a function of their preferred stimuli. And you can see, and the dots are the actual data. Again, the black line is the prediction from the theory that I just told you about. And you can see that it works pretty well. Obviously, there's variability, and everything is not lying right on the line, but it's a pretty good description. There's basically no free parameters here. So this is pretty good for a parameter-free prediction of what these cells should be looking like in this population. And again, just coming from environmental statistics. Right? I didn't use any physiology to derive that curve. I just used this. Okay, one last plot. On the right side, I'm going to be showing you perceptual results. So these are perceptuals as a function of spatial frequency. 2 different labs, 2 different sets of measurements, the blue ones and the red ones. They're different. Why are the numbers different in the 2 labs? Because they use different procedures. They had different stimuli. The size of the stimulus was different. The duration of the stimuli were different. They used a different criterion for the threshold. All of those things go into that constant alpha that was in the equation I showed you. But the point is that the shapes of the curves are the same, and they're well matched to what the theory said they should look like. Same basic slope and shape, i.e. almost a straight line. Okay, let's look at another one. So this is orientation, which is kind of what I was telling you to think about from the outset. That's actually a distribution of local orientations in photographs over a large collection of photographs. It's very non-flat. It's actually shocking to me how non-flat it was. There's a distribution of cell preferences near the fovea. This is a very old data set of cells that were recorded near the fovea. It's very difficult to record cells near the fovea. There are very few data sets, but I was really excited to hear Jack tell us that he's got tons of data now near the fovea. So I want your data. We don't know in this particular data set what the bandwidths look like, so I can't do this comparison. That's the prediction. We're putting it out there. There's the prediction, but we don't know actually what the data look like. But we do know, because we measured it ourselves, what human discrimination thresholds look like. And we went into this thinking they would be kind of flat or maybe a little bit off of flat, but they're actually remarkably far from flat, and they're remarkably well predicted by this theory. Okay, so those blue dots are actual data. The data were collected before we made the prediction, but by another person that I was working with. Anna Hirschig was a postdoc that worked with Mike Landy and I to do this experiment, but the experiment had actually a different purpose, but it turned out that it fit perfectly once we had this prediction put together. Okay, one more. speed, retinal speed. We don't actually know what the distribution of retinal speeds is in the world. People move their eyes and they move around while they're looking at things, and stuff in the world is moving, so it's pretty complicated to come up with actually a probability distribution of what local motion looks like on the retina. So we started on the right side. We start with the psychophysics. These are two labs, red and green points, red and blue points again. They've measured perceptual discrimination thresholds as a function of speed. And you can see they're both on top of each other in this particular case, and they can be pretty well fit by that curve. We take that curve and we pull it back. We say, where is speed represented in visual cortex? In the monkey, we believe that the best candidate for that is area MC. That's an area that's where all the cells are speed tuned and often direction tuned, and it's been well documented over many decades, and much of that work was done in Tony Mosh's lab, and he has a huge database of cells, conveniently so. So Tony gave us access to another data set, and this again works out pretty well. You can see that the tuning of those speed-tuned cells is pretty well predicted by the theory, and so is the distribution of preferred stimuli. We don't know what should happen back here in the world, because again, to measure this, you would need an eye tracker. You have to basically let a human or an animal move around their environment, do their usual thing, and you need an eye tracker, and you need to know exactly what they're looking at and how everything is moving on their retina. So there are people that are trying to do that. There are several groups that are trying to make measurements like that. It's pretty hard, but maybe at some point we'll actually be able to answer what this looks like. Okay. Oh, question, yes?
Eero, I'm trying to understand why for your middle row you're restricting it to neurons close to the fovea. Why are you...
Good question. Yeah. So there are known and pretty strong biases in terms of orientation preferences, depending on where you look in the visual field. And so we looked at a couple of different data sets. Some of them just sort of averaged. They didn't tell you what eccentricity the cells were, or even what angular position they were, and so they sort of mixed them all together. And we found that in those cases that there were these kinds of biases, but they were much smaller. They were flatter. I can show you examples of those. I think I still have them in here. But when we looked at the foveal data, we saw something that was as strongly biased as we expected from the theory. So I'm not sure what to do about that, to be honest. I think that foveal vision is kind of where we want to make these predictions, but I'm just hand waving about what I think is going on in this work. I think there are biases there, but we don't really know why. Okay. Thanks for the question. Two more examples. These are auditory. This is acoustic frequency. We took a large collection of natural sounds. We measured acoustic frequency in small bands. We fit that. Again, this is a modified power law, but it's much more modified than the one that I showed you for the visual case. This is an anesthetized cat, and it's, sorry, I have to look at my notes to remind myself. No, I didn't put it there. I'm sorry. I think it's inferior colliculus. I should remember this, and I forgot to write in my notes. I can't remember where these are recorded. If somebody needs to know, I'll look it up after the talk. Just check the paper. In any case, these are the density. This is the distribution of preferred stimuli, and these are the distributions of bad ones that we see in those data. There's quite a bit more spread than in some of the visual data sets that I showed you, but I think it's still pretty good. It's pretty well approximated by the prediction. And these are perceptual discrimination thresholds, again, human. Again, two different data sets from two different labs, and you can see that the predictions are pretty good. And the last one is modulation frequency. So you take those frequency bands and you ask if you measure power, local power of those frequency bands, and you look at how that modulates and what frequencies it modulates at. You can measure that in natural sounds. Again, you can fit that with something like a modified power law, and again, you can do a pretty good job predicting cells. And this, I think, is in auditory cortex. Not quite sure because I didn't put it in my notes. And again, you can predict human detection or discrimination thresholds pretty well. Okay, so you might wonder, so that's the set of all 5 variables I showed you. On the left, the environmental measurements. In the middle, 2 things about the physiology, and on the right, human psychophysics. And I think this is, I guess I would say this is a qualified success. but we need to look at a lot more examples and a lot more variables to really be confident about that. And one thing you might be wondering is, I started all of this saying that we were going to maximize information transmission, but maybe there's some, how do I know that that really matters so much? Maybe there's some other thing I could have optimized and it would have been just as good. Turns out that we can take this formulation and we can optimize lots of other possible functions of d squared g, that is functions of the Fisher information, including something that would basically try to maximize discriminability. So instead of maximizing information, what if you tried to just maximize or minimize, if you like, your discrimination errors across the entire range of S, the average discrimination errors? Maybe that's a better projective function, or maybe it doesn't matter, maybe it gives you the same answer. So we can go through and do that calculation. Again, nicely enough, we can do all this with paper and pencil because everything comes out in closed form. And when you go and you look at those predictions, they're not very good. So the green lines, I hope you can see them, the green lines are the predictions of what happens if you optimize for discriminability, average discriminability, and they basically don't fit the data. You can see throughout, in all 5 panels, there are pretty severe mismatches, much, much worse than the black lines that I originally showed you, which are still on these plots, just to remind you. Okay, so in the last little chunk of the talk, I'm going to shift gears. I told you I was going to talk about encoding and decoding. So what we have is a normative theory for coding efficiency that it turns out embeds the probabilistic structure of the world in the population. So that's the key thing. After we go through all this, after all is said and done, the distribution of those stimuli out in the world is now somehow embedded in the architecture. And it's in, if you like, it's in the synapses that govern the tuning curves of those cells. And they're arranged in such a way as to sort of reflect what is out in the world. So now the question is, on the decoding side, how do you make use of that? If I want to do decoding, I want to figure out what's out in the world, how am I supposed to take advantage of or make use of the fact that there's a probability there, there's a description of that probability distribution? And for that, we want to turn to decoding. We want to think about, well, Helmholtz first, who said that perception is a guess as to what's out in the world, given 2 things, our sensory input and our prior experience, that is what we expect to happen. And if we turn that into a Bayesian format, what he's really saying is we got some noisy measurements, that's our population representation. We want to combine that with some prior, some understanding of what things were like in the world. In my case, that's just the probability distribution. And from that, we want to make an estimate, that's our percept. So how do we do that? How do we do this calculation given this population I told you about? We know that there's lots and lots of cases in sensory systems, in sensory perception, sensory psychophysics, where people seem to behave as if they're Bayesian. That is, they're taking into account knowledge of the world, general probabilistic knowledge of the world, in making judgments and in what they see. And here is just a quick list of examples. There are many, many others, but there's a whole bunch of these. I've been involved in some of these, and many other people have as well. Okay, so we think that perception is kind of Bayesian, and that decoding is therefore sort of Bayesian. So how do we instantiate the priors? Bayesian requires that you have some prior probability distribution, or you combine that with your incoming sensory information, and that's going to allow you to make your judgments. Where's the prior represented? How do you acquire it, and then where do you put it? And so we're talking about memory, which I thought Randy would be pleased about. And people have proposed things over many years, and I think they're all reasonable proposals on the surface, but I think most of them in the end don't work at all. I think there's zero evidence. In fact, there's counter evidence for most of these, including some that I was fond of early in my career. There's the idea that we have some sort of weighting in a readout population. There's the idea that there are modulatory feedback signals, that there's spiking activity in some other population of cells that's kind of representing the prior, and we just have to combine that with our incoming sensory cells. There's the idea that it's built into dynamic network properties, and that the responses are samples from the posterior. That's an idea that Joseph Fisher and Pietro Bercas and Mate Lengyel and a bunch of others have been pushing. And the last thing is that the population is inhomogeneous. And these inhomogeneities are the things that actually represent inhomogeneities in baseline rates or gains or tuning widths or cell density. Something is capturing prior probability information. And that's the idea that I'm going to stick with. I just want to show you that it actually works. So it turns out if you want to do Bayes' least squares estimation, you have to compute the conditional mean of the posterior. You can write that down like this. This is just actually the integral over there's the prior, there's the likelihood, there's the stimulus. We're going to integrate that to compute the mean of the stimulus according to the posterior. We have to normalize this by that thing in the denominator to make sure that it's a proper probability distribution. We could write that out as a sum by taking these integrals and writing them in terms of a Riemann sum. And then we can think through, well, how would we do that if we were trying to represent that in terms of cells? And when you write out, using the model I described, how these probabilities are represented, they're just going to be exponentials of this weighted sum of inputs. So remarkably, when you actually work through this calculation, it looks very neural. So here it is. Here's the response of the population. I told you about the inhomogeneous population. We take each of those responses, we feed them into some exponentiator, something that is an accelerating nonlinearity. We take each of those and we take weighted sums of them, and we have to do normalization, so divisive normalization appears here, and then we take the weighted sum of all that, and remarkably, that little calculating unit calculates the posterior mean. So if we want to do a readout of this, it kind of looks like a population vector. It kind of looks like a weighted sum of the inputs, except that they have to get exponentiated and they have to get blurred or averaged with this filter W. And so people have proposed that you can use the population vector, the classic readout from Georgopoulos back in the 80s, to do Bayesian decoding. But it turns out that actually this is the right way to do it. And if you actually compare this modified population vector against a conventional population vector, and you measure the mean squared error as a function of the number of neurons you're using, and this is for 3 different total firing rates, that resource of total firing rate, these don't converge at all. They actually get worse as you add more neurons. They convert, they are very biased. And this modified version converges very nicely. So the performance is extremely good. You can actually use this to do Bayesian decoding. You can use that prior representation that is embedded in the population in a correct calculation that looks neural to actually do probabilistic inference and reasoning. So I think that that's pretty cool. And I'm going to finish by just reminding you what I told you. So these optimal populations, they give us a direct link between measurable quantities. So this is a somewhat abstract theory, but it actually comes back to Earth and connects to things that we can measure, but many, many people have measured. So that's why I was showing you data from the literature that's some of it's pretty old. These are things that people have measured for many decades. Environmental distributions, densities and tunings of neurons, and perceptual discriminability. I showed you that information maximization, which is what we were aiming for, works better, is a better description of the physiology and the perception than, for example, estimation accuracy. That's what happens if you optimize for discriminability. And I showed you that this gives you an embedding of environmental probabilities that can be incorporated into a Bayesian decoder. What's missing, well, lots of things are missing, but for me, the 3 things that are most important are, first, this was one variable at a time, and I kind of had to make ad hoc choices of what those variables were and which cells were coding for them. We want to figure out how does the brain decide which features to represent, and how does it do it for the multidimensional case, and how does it choose the features in the first place? Because, of course, once we go beyond V1 and early stages of any of the sensory systems, it starts to get very difficult to define what it is that's being represented, as we heard from Deb. And the last thing is, how does, I told you how I used a pencil and a piece of paper by candlelight in a blackened New York City to solve this problem. I'm exaggerating, obviously. But how does the brain do that? If you want to optimize, if you want to solve this optimization problem, something has to be built into the system to allow it to sort of regulate, calibrate, and learn how to get to these. And I don't think that the brain sits there and optimizes for Fisher information or any of the things that I told you about, but there must be local regulatory mechanisms that push the system toward these solutions. And we really have no idea how that would work or how to set that up. So I think that's an interesting problem. Okay, that's it. Thanks.